Okay. Kia ora koutou katoa. Good afternoon, everybody, and welcome to this week's Genomics Aotearoa seminar. Um, this week we have Mike Hoggard, who is a research fellow with Genomics Aotearoa and he is based at the University of Auckland in the School of Biological Sciences. Just a little bit about Mike. He actually started off as a philosopher. Well, he studied philosophy before finding his true love, which is biology, right, Mike? After finishing undergrad, he did a PhD with Mike Taylor, working on chronic rhinosinusitis, and won the Vice Chancellor's Best Thesis Prize for that. I was very fortunate then to have Mike come and join my lab to work on a project in an estuary, actually, so quite a bit of a change for Mike, working on viruses. And that's what Mike is going to talk to you about today. So, Mike, over to you.
Cool. Thanks for the introduction. Kia ora, everyone. So I'm Mike Hoggard and I'm based at the University of Auckland. As Kim mentioned, I've been here for quite some time now and currently a GA postdoc. So happy to share some of this work today. It's been a long time in the making, but finally happy to have had this published recently. So I'm happy to be able to share this with you as well. So a bit of background, a lot of this work was done under Genomics Aotearoa 1.0, the environmental metagenomics branch, and particularly looking at environmental microbiology. So that was the group that I was working in and the focus that I was mostly focusing on. So just a quick bit of background before I get into it. I feel like maybe this isn't an audience that I need to really sell this to, fortunately. But I just wanted to have a quick pitch for why care about microbial genomics. So microbes are really, really fundamentally important across all three pillars of the environment, primary production, and health. So microbes underpin food webs and ecosystems. They perform essential ecosystem services, really important for nutrient cycling, and also obviously across health and primary production as well. Actually, it's fundamental to understand microbial genomics for infectious disease, both for how infectious disease spreads and happens, and also for tracking. Also precision medicine. I think increasingly precision medicine isn't going to be only about the genomics of patients, but also the genomics and metagenomics of the microbes that they carry as well. And environmental microbes are also really important for biodiscovery of medically and industrial important compounds. So really, really important globally and important to know more about them. So today I'll actually be mostly talking about viruses of microbes. So again, just a little pitch. Why do we care about viruses of microbes? Well, all those important things that microbes do, viruses really importantly impact. So the microbial adaptation and evolution is influenced by viruses that infect them. Viruses also really strongly impact the turnover of microbial communities and the effect on ecosystem dynamics. And one really good example of this is in the open ocean. It's thought that up to a quarter of all bacterial cells are killed every day by viruses. And this affects the nutrient flux. So microbes are important for nutrient processing, but also it releases nutrients into the surface waters, which actually affects how much is passed to other trophic levels and also sequestered into the deep sea. So this has such a strong effect that it's being introduced into global carbon calculations because viruses have such a strong effect on this. There's also applied uses as well. So increasingly of interest, this has been around for quite a while, but it's increasingly of interest again, is phage-based intervention. So bacteriophage, viruses that infect bacteria. And the idea being here, if you have, say, bacteria that are of concern, whether they're a pathogen in human health or, for instance, plant health, could also be important for biocontrol, rather than necessarily using antibiotics, or maybe antibiotic resistance is a problem. If you can identify a virus that in a targeted way kills that bacteria, then the idea is to try and use that instead of a more broad spectrum antibiotics. There's also just an awful lot of them. So if we're interested in genomics, turns out viruses are the most numerically abundant genomes on Earth. So I feel like that's also just a nice added reason to be interested in learning more about the genomics of viruses. So with that quick little pitch out of the way, hopefully I've given you an idea of why I think it's important to know more about the genomics of microbes and also the genomics of viruses that infect microbes. Today I'm going to be talking about some of our recent work where we've been looking at the genomics of DNA viruses in particular. And I've roughly split the talk into three parts. The first part, we'll be talking about environmental niche partitioning of DNA virus communities. And I presented a little bit of the early work of this in a previous seminar quite a while back. So the first section, I'll just really be bringing up to speed about where we left off there, and then diving into some of the newer work we did on understanding the phylogenetic relationships of viruses across these environmental boundaries. And then the second section, we'll be getting into some work where we're looking at so genomic traits that are commonly associated with environmental adaptation in prokaryotes. We were then looking at whether we see these same traits in DNA viruses as well, as part of this niche partitioning, with a particular focus on GC content and amino acid biases, which I'll get into the details a bit later. And then the third section was trying to get into a little bit which tricky question of teasing apart if we do see these markers that are commonly associated with environmental adaptation in prokaryotes, if we see them in viruses, is the effect because they're actually directly adapting to the pressure from the external environment, or is it an indirect effect because they're actually just so strongly adapted to the host and the host is adapted to the environment? So it's trying to tease that apart, which is quite tricky, but we'll get into that a little bit later. So yeah, as I said, today we're looking at the niche partitioning and genomic markers of environmental adaptation in DNA viruses. At the end of the talk, I'll just touch briefly on where I'm hoping to go to next. It's not directly part of this work, but it comes out of this work, and that's trying to do a better job of identifying virus host pairs through metagenomics and through computational methods. And I won't have time to talk about this today, but I just wanted to flag, I'm also currently working on a project at the moment looking at bacterial CRISPR arrays and diversity and dynamics of those within closely related populations. Just wanted to flag that now. Hopefully I'll have a chance to present some work on that recently, because I think there's some cool findings that we're already seeing. So to kick into it, you might be wondering why I've been having these random pictures of estuaries on the slides so far. And that's because that's where this all starts. So this study first started out in the Waiwera estuary. So the sampling initially was 9 sites across the estuary, spanning from the freshwater zone all through the brackish mixing zone and out into the marine. So it's these 9 sites here. And that salinity divide was a really important part of the study design. And samples were also taken from water and sediment at the same time. And this was for metagenomics and metatranscriptomics sequencing. So within the group, there's a lot of other work that was going on looking at prokaryotes and eukaryotes, and with a particular focus on salinity adaptation and nutrient cycling. And you might recognize some GA postdoc alumni in here. So it's Dave and Annie and Boy, and this is all work done in Kim's group. So I guess amongst this, I came in and to find my own niche, decided to have a look at what we could see from viruses in the same data, same ecosystem, and ended up particularly focusing on DNA viruses. So there's this concept that's been kicking around for a while in microbial ecology. I just wanted to introduce it because it might be a bit distinct from eukaryote, what everyone's more aware of with eukaryotes, but it's perhaps a little bit contentious still, but the general idea is everything is everywhere, but the environment selects. And the idea underpinning this is that microbes tend to be dispersed everywhere. They don't have necessarily the same physical constraints on their dispersal that you might see with, for example, some terrestrial animals that might be constrained to an island ecosystem. And that island forms a barrier for dispersal, and diversification and evolution happens within that constraint. So the idea here is that microbes aren't constrained by dispersal, at least for the most part, and it's the environment that selects whether they proliferate where they are. So it's this idea of how the subsequent evolution and adaptation takes place and whether it's partitioned physically or not. And this is also relevant to viruses. So a recent study showed in a really cool way the same virus genomes and microbes identified across the natural water cycle. So they identified the same viruses and microbes from the ocean in through evaporation into the clouds, and then through rainfall back into terrestrial and freshwater ecosystems as well. So they identified this connectivity in the dispersal of microbes and viruses. So the question becomes, do they proliferate there? What does it mean? Do they land there and then just die out, or are they adding to that ecosystem as well? And so for the bit of background with prokaryotes, what's understood is this salinity barrier here is a really important physiological barrier. And so in this system, it's shown here, it's been shown elsewhere as well. For instance, when freshwater bacteria and viruses, this is an input into this system. They're traveling into here, but when they get here, you still see the genomes carrying out through the system, but the transcription just goes off a cliff. They're really unhappy, and generally speaking, they just die out of the system. So even though we have this dispersal into the system, they're just not adapted to survive in that environment. And this has been shown through particular adaptations that you can see markers of in the genomics of prokaryotes. So one of the questions here was, do we see the same niche partitioning with DNA viruses as we see with prokaryotes? And then if so, do the same adaptations, or at least the same markers of adaptation that you see in the genomes of prokaryotes, do we also see them in the viruses as well? So as I mentioned, the way we're data set was whole genome and whole transcriptome sequencing. There was whole genome assembly, prokaryote metagenome assembled genomes were recovered. And then also, when I came in as well, there was DNA viruses. There were tens of thousands of DNA virus partial genomes identified. 858 of them were over 50% complete. And in addition to that, so that was our initial data set, particularly looking at the salinity divide question. But we also wanted to see how many of our findings tracked into other ecosystems and also beyond just this estuary. So we expanded the analyses to look at the same questions in the IMGBR database, which is a database of globally distributed viruses. And we particularly focused on the high quality virus genomes, which are near complete. And there was nearly 300,000 of those. So looking at the same questions, but in a very, very big database of virus genomes as well. And just a few quick clarifications so I don't confuse things later on. All of the data I'm talking about is based on metagenomics and metatranscriptomics or genomic databases. So when I mention genes or protein sequences, they're predicted genes and proteins from the genomics data. And I also may sometimes use DNA viruses, bacteriophage, and cortivirocetes interchangeably, which isn't strictly correct. But cortivirocetes are the largest class of bacteriophage. Most bacteriophage fall into the cortivirocetes, and most DNA viruses tend to be bacteriophage. And it just happens that most of the data we have and also in the databases are cortivirocetes. They're just the most abundant, at least, that we know about and that are represented in databases. So the analyses, for the most part, were on DNA viruses as a whole, except for a few sub-analyses. But I'll often be talking about bacteriophage or cortivirocetes because I think the findings are most robust for them because they represent most of the data. And also occasionally I might use hosts, prokaryotes, and bacteria interchangeably, so putative hosts of the viruses. That's because a lot of the comparisons we're using prokaryote metagenome assembled genomes from the same data. And it happens that they were all bacteria. We didn't have archaea in there. So in these analyses, I didn't look at potential eukaryote hosts. So there could be some confounding things in there. But most of the time, I may use that interchangeably, sorry, but a lot of the time it's bacterial data I'm looking at. So just a quick background on the niche partitioning work. I introduced this before, so I just want to cover the main takeaway really was that what we're looking at here is each point is an entire community. And so the main takeaway is that communities from brackish and marine sites tend to look more similar to each other, but they look different to ones from freshwater sites, which looks different to sediment sites from the freshwater zone and different to brackish and marine sites from the sediment as well. And these same distinctions carried over into our work with the DNA viruses as well. So we see the same niche partitioning at the community level. And we did a lot more analyses to look into this as well and found that the DNA viruses tracked really, really closely with changes in the communities, I should say, tracked really closely with changes in the bacterial communities. There were a few residual effects from some environmental parameters, or at least something that co-varies with those environmental parameters, affecting the DNA virus communities as well. But it's important to note that it's really hard to tease apart what the direct effects of environment might be on these communities of viruses, because their hosts, they're obligate parasites, so their hosts are also really structured by the environment. So it's really hard at this level to tease those two things apart. So given that these boundaries exist, and it's been seen in other data sets as well, viruses have been seen to be stratified by temperature, salinity, oxygen concentration, broad ecological zones in the open ocean, and also between aquatic and terrestrial viruses. So the next thing we wanted to know was, given these this niche partitioning, what is the phylogenetic relatedness of viruses across these environmental boundaries? Do you see differences in the communities, but actually the viruses are still really closely related? Or is this, say, transition into these different environments more evolutionarily ancient? Does it go further up the hierarchy of taxonomic ranks, and you don't see much speciation happening across those boundaries, but you do within the boundaries? As I mentioned, we looked at this in the Waipapa estuary, focusing on the salinity divide, but then expanded it also into the IMG via database, which is global, and mostly looking at distinctions between freshwater, saline, aquatic ecosystems, and terrestrial. So getting into the DNA virus phylogeny, unfortunately, it's still quite a messy affair. There's no single gene that you can use as a proxy across DNA viruses, like you have with, for instance, 16S RNA gene in bacteria. RDRP is used commonly in viruses, RNA viruses, sorry. Gets a bit messier once you get into eukaryotes, but ITS and 18S is commonly used for fungi. You might use CO1 for other eukaryotes. It doesn't work for DNA viruses, though, because they don't have a conserved single gene you can use as a proxy. Also, even though the genomes are reasonably short, you can't do whole genome alignment either, because there's so much gene transfer going on and rearrangements within the genomes. So in light of that, two approaches have been developed. One looking at concatenated core proteins phylogeny, which I'll get into in a second, and another one doing gene sharing network. So the idea being the more genes you share, the more likely you are to be closely related. So getting into the concatenated protein phylogeny, this is a bit similar to doing a phylogeny based on one gene. But the idea here is identifying a suite of relatively conserved genes. So there might not be one conserved one, but there might be a set of relatively conserved ones that you can do a concatenated alignment of the proteins and use that as a proxy to infer the phylogeny of the viruses as a whole. So this has already been applied for bacteria and archaea using GTDB. And here, Phil Hugenholz's group came out with this paper a couple of years ago, applying the same principle to cordyceps, so the bacteriophage class, or one major class of bacteriophage. Unfortunately, with this new database updates means we couldn't just take the same core set and apply it to our data. So we worked through the same process again as they outlined to identify a new set of putative core genes or core proteins for the cordyceps class, and then applied that to our data as well to assess with the niche partition results. So this tree here we're looking at is a phylogenetic tree of the viruses in the RefSeq database, as well as our Waipapa viruses. Key takeaways here are firstly that this tree, so this is based on that core genes approach, roughly fits quite well with the current taxonomic affiliations of the RefSeq viruses. And the second thing being these spokes here are our Waipapa viruses, and they tend to fall within clades that are already in the RefSeq database. So this is quite common for aquatic viruses. They seem to be increasingly well represented to some degree anyway in databases. But recently it was shown, for instance, that terrestrial viruses are still quite poorly represented in these databases. Looking at the niche partitioning results now, the key thing here is just the darker colors are from the saline viruses from the saline environments, the lighter colors viruses from the non-saline. First thing being that, as you can see, all of these environments based on salinity, so non-saline, saline, are spread throughout the tree. So we don't have a case where one whole side of the tree or a whole bunch of clades are found only in saline environments and the other side is non-saline. It's really interspersed throughout the whole tree. But the second thing is once you get down into subclades, so more closely related viruses like we see here with shorter branching events, it's very, very rare that we saw representatives of viruses from across that salinity divide. So most of these subclades were either only saline or only non-saline viruses. And when we extended that into the IMGBR database, so in this case, this is tens of thousands of cordyceps viruses, so the tree's a bit of a mess to look at here, but you might just have to take my word for it that it's the same principle fell out of this, that looking at aquatic saline, aquatic non-saline, and terrestrial viruses, we saw representatives throughout the entire tree, pretty equally interspersed. But when you get down into those more closely related subclades, it was very, very rare to find viruses jumping across those three environment types. So key findings there, at least for the method, using this core genes approach to infer the phylogeny of the viruses, there was overall agreement with the current viral RefSeq taxonomic affiliations. At higher taxonomic ranks, tended to see viruses represented across these environmental divides. So, for instance, at say maybe phylum and class, they tended to be across the environmental divides, but in subclades, they tended to contain only one ecological niche type, so either non-saline, saline, or terrestrial. So the second approach for this is referred to as a guilt by association method. So the idea being this tool builds a gene sharing network of all the virus genomes based on the principle that the more genes they share, they tend to be more closely related. So that's the idea underpinning this. This is a tool that's been around for a few years now. And you'll have to excuse the Jackson Pollock-esque figure here. You don't have to take too much of the details, but the main thing to know is that each point is a individual virus genome, and the edges, so how they're joined, is based on how many genes they share. So it's their gene sharing, and they're colored here by environment type. One thing within this tool is it includes a clustering process. So it clusters virus genomes based on their gene sharing. And if they fall into a cluster, based on the parameters that are built into this tool, they tend to be from roughly the same genus. If they don't fall into a cluster, then they're not from the same genus. So looking at these clustering analyses, here we're looking, so these are cluster numbers. So again, this is approximates individual genera. And what we found is that for these clusters, roughly over a quarter of them were only had representatives of viruses from freshwater environments. Roughly a little over a quarter of them only had viruses from saline environments, and over a quarter of them roughly only had viruses, sorry, not roughly, but only had viruses from terrestrial environments. So we see this really stark partitioning. So that's approximately equating distinct genera, and they had no representatives that crossed the environmental boundaries. These handful of mixed ones in here, when I filtered out possible edge cases where, for instance, like nearshore environments or estuaries where you'd expect distinct ecosystems to be, viruses from distinct ecosystems to be recovered. When I filtered those out, there were very, very few of these clusters left in the middle that had any crossover between the groups. So as a takeaway there, genomes in the vContact2 clusters, which approximate virus genera, often only came from a single ecosystem type. So that was non-saline, saline, or terrestrial. So bringing all this together is just a very rough schematic, but just to give you an idea of what we're looking at here, the idea is that it looks like DNA viruses at higher ranks tend to be from across these three ecosystem types. Tend to be a bit agnostic of that, and whether that's that they've evolved in parallel across these types, or they evolved in one ecosystem type and then diversified out of that. I'm not sure, but at these higher ranks, they tend to be across the ecosystem types, and this includes really major morphological changes as well. But then once we get down into approximately around about genus level, it's very, very rare to see representatives from the same genus across these distinct ecosystem types. So it seems like the diversification happens once we get down to about this level. They seem to be relatively constrained to those ecosystems. So there is almost like an island effect that happens there where they're constrained to within the boundaries of that ecosystem. And then all the diversification is happening within that. And it's very rare you get jumps into these other ecosystems. And interestingly as well, this actually tracks quite similarly with what's known about prokaryotes as well. There tends to be, it's quite rare to find species from the same genus across particularly the salinity divide, but you can see genera from the same family across that divide. So it's interesting that it's kind of a similar threshold there. So now that we've got that far, we are seeing these distinctions in niche partitioning. So the next question that came up for us was going back to this question here about everything is everywhere, but the environment selects. The question is for DNA viruses, does the environment select? And what is the important environment here, I guess, is also a key thing to state. So as viruses are obligate intracellular parasites, there's a strong argument to be made that inside their host cell is the environment that matters to them. But viruses that infect prokaryotes, they do also, a lot of the time, the first part of the infection cycle does actually happen outside of the cell. So they are also exposed to the environment. So the next question was, do they also have some adaptation or selective pressure based on the actual physical constraints of the environment, and does that play into some of this niche partitioning that we see? In bacteria, environmental adaptation has been quite well studied. GC content is often associated with different environment types. Historically, it was associated with salinity, particularly from studies in hypersaline environments. I put a question mark there because it seems like maybe this actually tracks with something else that's characteristic of hypersaline environments as well, because there's been some differences in other saline ecosystems, but that still tracks often with environment partitioning. And the second one is amino acid biases and protein acidity. So this one's been really strongly associated with salinity specifically, where bacteria will, and you can see markers of this in their genomes, they'll up the use of acidic amino acids in their proteins to up the protein acidity, because this has important effects on the protein structure and function in saline environments. So they tend to fall apart and not work as well if they don't have these adaptations. And in bacteria, there's this split between the approach of salt out versus salt in. So in environments similar to estuaries and marine ecosystems, the level of salt that's there, they tend to be able to pump that back out of their cell. So the intracellular environment tends to be not to need to adapt so much for the salt, but anything on the cell surface or secreted outside the cell, you tend to see these adaptations in the proteins more. Whereas once you get to hypersaline environments, they can't keep up with that. So you tend to see the adaptations required for the protein function throughout the whole proteome, which you see in markers throughout the whole genome as well. So we hypothesized that at estuarine and marine salinities, you might see the same distinction in viruses that infect bacteria, because the first stage of the infection cycle happens outside the cell. We hypothesized that the proteins that are exposed to the external environment might be under the same selective pressure, and that might be different to the proteins that have to function inside the host cell. So, for instance, one's to do with assembly of the protein or replication. So just to give you an idea of the stereotypical cordyceps virus, we've got this capsid, the genome's contained inside, and then we have this tail, which has different morphologies in different groups, and that's important for the first stage of the infection. And then these tail fibers and the spike protein are important for host recognition and binding. So the idea being that these ones may tend to be under more selective pressure from direct environmental physical constraints than the internal proteins. So the proteins that are expressed internal to the host cell. So first looking at the GC content. Here we're looking at all genes, and just to break this down, salinities across the bottom here. So from the freshwater out into the marine salinities, and the GC content here. So looking at all genes, we saw this consistent trend of reduced GC with increasing salinity. And when, oops, sorry, I went the wrong way. When we looked at the other subgroups of genes and slash proteins, depending on which analysis we were doing, we saw the same trends. So for the quote unquote internal ones, we looked at large terminase and DNA polymerase. The external ones, we looked at some of the tail proteins, the capsid. We also looked at endolysin because it's at this interface where it has to function at the cell wall. And we consistently saw the same trend of reduced GC with increasing salinity. And this mirrored quite well with the prokaryotes in the water column as well. Interestingly, there was a bit of difference in the sediment, which needs a bit more work to understand. But it seemed to be quite consistent and consistent with what we saw in the prokaryotes, which are the putative hosts, at least for any bacteriophage. And we also saw this when we looked at the global data set. So here, the median here, the dotted line, that's the median of the non-saline group. And then we saw this reduced GC in the aquatic marine viruses globally. But then we actually saw this elevated GC in the hypersaline. So interestingly, we see GC effects with environment and these environments that we're defining based on salinity. But it seems like maybe it's not a adaptation due to salinity. It might be an adaptation due to something else in the same environments. And again, we actually do see similar things with prokaryotes as well. And then in terrestrial, again, elevated GC, and again, we see that in prokaryotes. So it looks like quite consistently that this is across the entire genome. So all the genes in the genome. Doesn't seem like there's a selective pressure happening for the external proteins versus internal proteins on GC. And it seems to mirror quite well with the prokaryotes. So takeaway there is it seems like it's very likely re-inherited from the hosts rather than a external adaptation. I also find this interesting as a bit of an aside that even though it might not track salinity adaptation, we do see these differences between ecosystem types. And I think that could be interesting if we're moving forward into things like monitoring, like real-time environmental monitoring, whether things like this can start going into predictive models, so we can identify changes in the environment as well based on things like GC, not only GC. And obviously this doesn't explain everything because there's a lot of overlap here, but maybe it's a value add. And for instance, if you were looking at rivers or neutral environments, do you see the changes in GC if you have a heavy rain event and you have a lot of sediment influx into those environments? And do you see differences based on the land use change around those environments as well? So I think there's interesting things you can take away from that. That's, as I said, a bit of an aside. So the key findings there, reduced GC with increasing salinity in the estuary data. In the IMG-BR database as well, we saw the same trends, but then we actually saw increased GC in hypersaline. So it doesn't seem like it's a salinity response as such, even though we see differences in these environments. And the effect is seen across all genes. So it doesn't seem like it's selective adaptive pressure, at least not from the environmental questions that we were asking. It's very likely a selective pressure from the host. So now getting into the amino acids. Again, here we're looking at the Waipapa estuary data. Just to clarify here, what we're looking at is, as I mentioned before, the adaptation is increased acidity, increased use of acidic amino acids in the proteins, because that affects the protein structure and function in cellular environments. So here we're looking at the proportion of acidic amino acids in the predicted proteins minus the proportion of basic acidic amino acids. So the idea is the higher this is, the more acidic the proteins are. So here, looks like a flat line basically is there is a very subtle increase in protein acidity in all proteins with increasing salinity there. But then when we look, other way. If we look at the internal proteins, proteins that function inside the host cell, similar trend. But once we start looking at the ones that have to function outside the host cell, we start seeing this trend of increased protein acidity with increasing salinity, and it's by far the strongest in the Metagenomics protein. We still see it in the major capsid and the endolysin, but as you can see there, if you can see the R squared, very small effect sizes. These aren't too dissimilar to what you actually see in prokaryotes as well, but we by far and large see the largest effect here with the major tail, which I think functionally, because of its role in infection and also the need to have functional conformational changes during infection, I think there's also a pretty strong explanation for why that would be the strongest affected here as well. So we see the subtle increase overall, but it's more pronounced in these quote unquote external proteins, the proteins that function outside the cell, but in contact with the external environment. And then this is just to show that we saw the same trends in the global database as well. So strongest for major tail, and still saw a trend, but it was less pronounced for the rest of the proteins that we looked at and the proteins, the overall proteome or predicted proteome. Also, just to give you a bit of a visual idea of what this looks like, and also because this was requested by a reviewer, but it gave me an excuse to have a play with AlphaFold for the first time, which I wasn't complaining about. So these here are based on the predicted protein sequences put into AlphaFold to predict the protein structure. And these are actually multi-protein complexes of the same protein, but there's between 12 to 18 of them in these. This is the major tail, and they form these tubes of this multi-protein complex. This white area here is one of those proteins, and the red gives you an indication of the electrostatic potential, which is directly related to the acidic amino acids on the surface of the protein. So it's a proxy for the protein acidity here. And electrostatic potential is really important for protein interactions. So we can see here in the non-saline environment and then going into marine and hypersaline, this more red means more acidity on the protein surface. And then we have the same thing. These are hexamers of the major capsid protein forms these hexamers and tiles, which fills the whole capsid, and sometimes pentamers, but these are the hexamers. And we see the same trend. So you can visually see this increase in electrostatic potential, which is related to increased surface acidity of the proteins. So key findings from this protein acidity aspect: there's a subtle increase in protein acidity overall with salinity. And I didn't talk about it actually before, but we did have an indication that it seems like the most important thing here really is the non-saline to saline transition. We didn't see much of a relationship with increased salinity being more acidity. It really seemed to be the difference between non-saline and then saline environments, but for instance, hypersaline environments didn't look especially different to marine environments in terms of their protein acidity, which was quite interesting. So it may be that DNA viruses are a bit more robust across that salinity gradient when they're in the saline environment, but there's a strong divide between those two. And importantly, for our takeaway, while there was a subtle increase across the board, it was particularly pronounced in those proteins that have to function outside the host cell, and especially the major tail protein. So moving on from there into this last section, that led us to wonder if we do see these markers of genomic traits that have been associated with environmental adaptation in prokaryotes, if we also see them in viruses, is it a result of actual direct adaptation or adaptive pressure from the environment on the viruses, or is it that the hosts that they're infecting are subject to that adaptive pressure, but the viruses are actually just purely adapted based on their host. They just inherit these traits from the host, but they're not actually there's no direct adaptive pressure on them. And this is a really tricky thing to pull apart, and especially because, as I mentioned, viruses are obligate intracellular parasites, and they rely, for the most part, on their host for the translation machinery that encodes their proteins. And most of their life cycle happens in the host. As I mentioned, not all of that, but so that's really, really strong selective pressure. So the question is, does the environment add to that, or are they actually just inheriting that from their host? So to look into this, in one way, we looked at what the patterns were of host tRNAs. So host tRNAs, as I said, the viruses for the most part rely on their hosts for the translation of their proteins. And we were looking at patterns in tRNAs for particular amino acids relative to the proportion of amino acids that we see in proteins for viruses, and also the putative host. So in this case, again, that's looking at bacteria in the estuary. So in these, just to break this down quickly, sorry, I didn't label the x-axis here, but this is salinity again, going from 0 up to the marine, so 32-ish. And the blue here is amino acid proportion of glutamic acid in virus proteins. Orange is the bacterial proteins, and the red here is the transcription of the tRNAs in the bacteria. So for instance, in glutamic acid here, we see there is this trend of increased tRNA transcription of glutamic acid, but it wasn't significant. Whereas we do see a significant increase in the glutamic acid amino acids in the proteins of both the bacteria and the viruses. And the rate of increase in these was I can't remember exactly, one to two orders of magnitude more than the increase that we did see in the transcription as well. In aspartic acid, which is the other acidic amino acid, there's actually a really marked disjoint here between an increase in aspartic acid in the proteins of viruses and bacteria, although at different rates, but a significant decrease in the transcription of the host tRNAs. When we looked at the basic amino acids, arginine, histidine, and lysine, again, particularly for the viruses, there was this disconnect between the trends we saw in the amino acids that they actually encoded for in their proteins and the transcription of the tRNAs that were ostensibly for those amino acids. And we also looked at alanine, because that was another important amino acid in the story. But again, there was disconnect between the patterns we saw at the protein level versus the translational machinery of the hosts underlying that as well. So I'm just going to skip through these figures, but because it's just presenting the same data in another way. But I just wanted to focus on the takeaways here, being that from our analyses, and we did a bunch more as well, looking across not just the acidic and basic amino acids, but across all the amino acids, it was pretty consistent that the host tRNA abundance and transcription didn't explain the amino acid biases that we saw in the hosts, in this case bacteria, or the viruses. So there's this disconnect between what we saw in the ostensibly the translational machinery for proteins and what we actually saw in the proteins. So it's not a simple story where you can just look at the post-translational machinery and the viruses are adapting directly based on that. And for glutamic acid specifically, that was one exception where we often did see significant association between the transcription of host tRNAs and both virus and bacterial proportions of the amino acids. So in this case, it's glutamic acid here, but in both cases, the quote-unquote external proteins that we expect to be more influenced by adaptation to the external environment, so the major tail here, and in the bacteria, this was a secreted protein, so proteins secreted outside the cell. In both cases, that trend was no longer significant. So there's obviously some additional effect here. It seems like maybe there is across the board an association for glutamic acid with the host translational machinery, but something else is going on and influencing, by the looks of it, this major tail and the secreted proteins for the bacteria. So again, gives us an indication that there's more than just that sole adaptation of viruses based on the host translational machinery. So to summarize, distribution of DNA viruses and bacteria are similarly constrained to specific ecological niches. And from that, it follows that proliferation of viruses transferred into distinct biomes, at least based on non-saline, saline, and terrestrial, are rare, because otherwise we would have seen them in all places in these analyses, but we didn't. We saw quite distinct differences between non-saline, saline, and terrestrial. Therefore, they clearly don't proliferate very well, even if they are transferred between these different environment types. We saw that diversification within approximately the level of genera was restricted to these niches. So it was very, very rare that we saw representatives from the same genus across these environment types. So it seems like there's a pseudo island effect going on here for the ongoing diversification and evolution of these viruses. Adaptations to salinity were also present in viruses, or at least the markers that we see in the genomes of prokaryotes, we also saw them in viruses. And some of these adaptations may be due to the direct influence of the external environment, and that's based on the fact that we saw this especially strong effect, particularly for the major tail, but also for the major capsid protein as well, in increased protein acidity. And again, as I mentioned, these are proteins that have to function outside of the host cell, so they are in contact with the external environment and have to be functional in that environment. So we think there's good evidence there that there is likely some environmental adaptation effect going on for viruses as well. One important caveat there, as I mentioned before, hosts are under extremely strong adaptive pressure. Viruses under extremely strong adaptive pressure based on their hosts. So this includes the host translational machinery, host defense mechanisms, various protein-protein interactions. They're also under structural limitations. For example, they have physical constraints on the capsid size and architecture. So those are really important adaptive pressures. And the question here is, are there also additionally some adaptive pressures from the external environment on top of that as well? So a couple of unanswered questions that remain. We started looking into this, trying to tease apart this host versus environment question about what the viruses are adapting to. Are they getting these traits of environment adaptation because of direct adaptation, or is it indirect and they're actually acquiring them via coevolution with the host? We started teasing that apart, but I think there's more work to be done there, but it's quite hard to pull those two things apart. I think there's some lab experiments that could be done, but it is a very tricky thing to pull apart because of the obligate requirement of viruses to replicate inside their host. And also the next question being, if they do experience direct adaptation based on the environment, is this biologically meaningful? Or are they so restricted based on their host range, and hosts are so restricted based on environment type that it doesn't matter? So is this a biologically meaningful additional restriction on virus proliferation beyond the strong host range limitation already? So that's still an ongoing question as well. And as I mentioned, I'll just touch on future directions. So out of this, one thing that came out, it's not a feature of the main publication in the end, but in all the work I did across a lot of different tools, I found them all pretty poor with identifying virus host pairs. So say viruses and the bacteria in the same data that they're likely to infect based on genomics data is still really hard to do. So a next step that I'm hoping to get off the ground is a couple of different ways of trying to tease this apart. One is an idea I have going, looking at new AI models based on protein structural prediction and interaction to try and see if we can computationally predict the interaction of the host receptor sites and the the attachment proteins between virus and host, and see if we can predict the likely host that viruses can infect that way. And another way is through single-cell genomics and many metagenomes. And this is a bit of a guilt by association thing. You see them a lot of times together in single cells or many metagenomes, then they're more likely to be a virus host pair. And this is really important for understanding how viruses are actually infecting individual bacteria in any setting. If you're interested in any resources, there's a lot of these resources are in the GA GitHub under the environmental metagenomics. Also, the summer school has a lot of this. The whole workflow behind this particular study is also on GitHub as well, including all the scripts and everything that went into it. And I'm happy to say, as of last week, this is all also finally actually published as well. So if you're interested in reading any more detail or just helping our metrics by clicking download, feel free to go and find that as well. And with that, thanks to everyone in the Handley group. And then there's a lot of people involved in the Waipapa study specifically as well. And also thanks to Genomics Aotearoa. Been very fortunate to be a postdoc with GA for quite a while now, and also for University of Auckland, where I've been parked up for a long time now as well. All right, thanks.